Now we are going to get to what we call cool science. And in particular, what I've chosen is sort of 4 or 5 cool science, where we need to recall what storm and palm are. That is, the normal resolution that you can get with fluorescence microscopy is what is the resolution? Come on. Right, and what is that? That's exactly correct. And what is that? How many microns or nanometers or angstroms or whatever it is? This is a fluorescence technique, and
it's the diffraction limit of light.
It's the diffraction limit of light, which is about what? What's that? Lambda over 2, and lambda is the emission wavelength. So lambda is 500 nanometers. That's bluish green. So it's about 250 nanometers. And if you want to see with super resolution, that is, you can see much better than that, you need to use one of these new techniques that just won the Nobel Prize, which is either called storm or palm, and we'll go over that for just a second. There is actually another technique, a super resolution technique, which is called STED, and in fact, that also won the Nobel Prize in 2014. And that STED is really an amazing technique, because it's amazing because you label something with fluorescence. That is, you label something with an individual or a few fluorophores, and you actually make the thing laze. You actually form a laser out of this individual or a couple of fluorophores, and we'll see how that gets you super resolution. Okay, then we'll talk about EM, electron microscopy. Basically, this technique, as in every other technique, is limited in general by lambda over 2. So one of the things, if you make lambda much shorter, you can get to better resolution. And we'll see how EM does that. And you can get some amazing pictures, which we'll show. Then, in fact, EM and X-ray together is a super powerful technique. It overcomes essentially both of these problems, and we'll discuss how that is. And I should say, if I were to start over my career and work on a new project or something like that, I might indeed do this combination of EM and X-ray diffraction. So I'll try to give you a sense of why I'm so thrilled by it. Okay. This is cool. Remember we talked about green fluorescent protein, a protein that will glow green, and that initially they were found in jellyfish, and now they've put it in tobacco and even no, right there. And even monkey, and I said, a mouse is a really good model system. Okay. Wait, there's no sound.
Red one in November. So the green one and a red one.
Oh, gosh.
Yeah, I think there is sound. It's just really coming out of there.
And you can have, they have the blue also.
You do?
The blue isn't very bright, so there's no sort of impetus to try and make a blue one.
Yeah.
So it's not easy to make the first one. He's fertile.
Okay.
So if I have a male and female,
Right.
we had. So I'm getting a red one in November. So now I have a green one and a red one.
Yeah, then. But you can get, and you can have, they have the blue also.
The blue isn't very bright, so there's no sort of impetus to try and make a blue one.
Blue fluorescent
So
protein.
it's not easy to make the first one. He's fertile. So if I have a male and female, we had them for a while.
He's fertile.
You get these tiny little like jelly beans and they glow. But she ate them.
Anyways, the point is you can make a green fluorescent mouse.
No.
Yeah.
When they say he's fertile, would he pass on the green fluorescent protein?
He's nocturnal. So
You know?
Yes.
That's awesome.
Are they sending any light on it or just dark and it's just
No, they're shining light because the thing is green fluorescent protein, so you have to shine light, and then it will emit at green. So it's probably they're shining UV light at it, so you don't see that per se.
how did they get the fluorescent blue? Change it from green to blue in the video?
I don't actually know. Okay, so this is just a review of what storm and palm is. And remember, there you have an object and you label it with a special fluorescent object, can be a fluorescent protein or not, where initially it's completely dark. You go and you shine typically a UV light on it, and you activate an occasional fluorophore. Now you have them fluorescent. You shine regular light, visible light on it. Because they're individual ones, you can get the point spread function of that. You can determine with Fiona where the center is. You can get it such that you know where it is to within a nanometer or so. You record that, then you do it again. And in general, a different one is fluorescing. And so you do it again and again, and eventually you can get your nanometer type of resolution. So this is the normal resolution, and this here is the storm resolution. Okay? So that...
No.
Well,
in the box?
there. Yes.
This storm give a lot of
Go ahead.
calculation and post processing system. So how long does it take to take 1 minute?
The storm in general is a slow technique. That is, it can take like a half hour to develop this entire thing. Yeah, it's a lot. They've done, in fact, efforts to increase the speed, and in some cases they can get it where you can actually get the whole thing in like a minute or two. Okay, yes?
There are two questions. Can this storm be done on any normal microscope or there are specialized microscopes?
Okay, can it be done on any microscope? Well, any microscope is a bit much. First, you have to have the fluorophores to label them with. Then you actually have to have a laser, you know, with enough power and so forth. And the big thing is your detector should be very good in terms, you know, collecting all the photons.
And does storm use phenomena in its processing?
Yes, because basically to get the to know where this molecule is, it's normal fluorescence, but then you get nanometer accuracy because you determine right where the peak is. That you get with nanometer accuracy. So that uses the Fiona technique. Yes?
Since storm is slow, is it usually used on fixed cells instead of black cells?
It is essentially always used on fixed cells. Yes. Okay? Okay, so this is all material that, you will know for the final. Okay, and you can get some amazing pictures, and this is, for example, at the end of one nerve, and this is on another nerve, and they're not quite touching. In fact, there's like a 40 nanometer difference between them. And you release some glutamate from this side. The glutamate, which is in little vesicles, binds to the membrane, releases. We now know by diffusion how fast it goes across, and then interacts with the second neuron. And these synapses are really important. You have about 100 billion of them in your brains. Actually, no. Synapses, you have about 100 trillion of them in your brain. Okay. So that was the method of storm, also called PALM. Now I'm going to tell you about another mechanism which I think is really cool, which is to get high-resolution fluorescence in a totally different way. You collect your fluorescence there's a certain width, again, it's like a mountain, a certain width, but if you have enough photons, you can get it to 250 divided by the square root of n. This is well known. So that's one way of determining where the center is. Another way is narrowing, making this have a smaller width. You can do that by going to the UV, making lambda smaller. The problem with that is you tend to kill samples. So this is now a very clever way of using visible light to do the microscopy, but yet managing to somehow get this smaller. Okay, so can you shut off the light? Thank you. So this here is an energy state of a fluorophore. You come in with this wavelength, let's say in the green, and you excite it. You excite it in general to some excited state that also is excited vibrationally, and it loses the vibrational energy within a matter of picoseconds, and then it comes here, waits around for about a nanosecond or two, and then in general it fluoresces over here and then loses its energy. And again, the difference in wavelength between the excitation and the emission is called the Stokes shift, and that's really good because this is in general red and this is green, for example. And so you can put in a filter over here where you come in with the green, and this is called a dichroic filter. It reflects the green. You have your sample, which is fluorescent. It emits red, and then it goes back and comes through this and is completely separated from the green excitation. Okay? And then, so what you have here is you have a normal excitation, and you get this mountain peak. Now, let's say that you not only have the normal fluorescence, but let's say when it gets here, you shine an intense amount of light. So intense that when the molecule sees it, it says, I give up, I'll stimulate, that is, that light will stimulate the transition to go back down. That stimulation, what does laser stand for? It's an acronym. Who knows what it stands for? Maybe, what do you think?
Like light amplification sense emission?
Stimulation?
Light amplification by stimulated emission. Exactly. That is, if you put an object, make it in an excited state, the probability of it coming down is a function of its chemical environment, but also a function of the number of photons around it. So you've put in a huge amount of light, and that will come in and force the molecules down. Now, if you're clever about it, what you do is you have it look like this. It's spatially sensitive. That is, you have it such that there's zero light right in the center. It's essentially a donut. And then a lot of light right over here. So you have the excitation light that's on. You have your stimulated emission that's also on. The combination of the two, what happens? Right over here, it's not having any effect at all because there's no light. So, in fact, it emits normally. All the excited molecules, which are here, are stimulated, come down, and so you don't actually ever see it. And so the result is that you get an effective PSF, point spread function, that is much smaller than this distance, which is lambda over 2. And in fact, how much smaller? Well, it can get down to like 6 nanometers. 20 nanometers is more common, or actually the truth is 40 nanometers is more common. Still, that's a factor of 5 or 6 better than 200 or 250 nanometers. And this idea won Stephen Hell, the person who invented it. And in fact, it does work, that in fact you get something like 190 nanometers. That's what they call the confocal. We won't worry about it. And with STED, we get on the order of 35 nanometers. Yes.
I just want to make sure I get it. Will you explain one more time really quickly how
Sure.
you get the really tiny dot?
Okay. Go ahead. You explain it to me.
So there's the original excitation band that you have. That's the green thing, and then you get that step pattern which you explained, and then they interfere, and then you only get the dot in the middle. Is that what happens? I feel like no, because of that silence.
What do people think? Okay. I'm gonna take off. You're gonna have to explain it. I'll be back in 3 minutes. So bye. So you can lead the discussion. Yeah, I know. Go ahead. Stand up.
Who can tell me how it works? Sorry, everyone.
Josh, let them struggle. They're ready? You ready?
Well, we got an idea, I guess.
Okay.
Basically, we thought that the bottom mirror focuses the two lasers onto the sample, but then this top mirror, when they come back, filters out the combination of the two lights, and that combination that's getting filtered out is like everything that like used to be around here, which is why the only thing that gets let through is the green that was going through the hole in the red light.
Okay. No. Just stay there. Stay there. So the two mirrors, you're talking about these?
Mm-hmm.
Okay. And what do these do? So just start again.
What we thought is that this bottom mirror sort of focuses the two lights together onto the sample through the objective.
Yes.
And then, yeah, okay. And then up through the detector, this top mirror, what we thought it's doing is it's filtering out through some mechanism, the combination of the excitation laser and the STED laser, so that you get this effective PSF of just the center of the excitation laser.
Okay, so what these colors are supposed to tell you exactly how that's happening. Okay, so this mirror, it is doing what? It filters out. Well, maybe filters out. What does it do? What does it reflect?
Excitation.
So let's say the green is excitation and the red is the so-called STED. Okay, so it's being reflected green. So everything green and shorter or whatever, green is it's reflecting. Okay? So it goes in the sample. The green is transmitted just fine. And what happens? You get some fluorescence. Okay? And then that fluorescence is what? So let's say orange. So the fluorescence comes back collected by the objective and then is going straight. So what does this do? Does it reflect it again? Or does it do something else? It's just passes through. I mean, it doesn't do anything to it. So this is wavelength dependent. Okay? So now it's just going through. And in this case, it also just goes through. Doesn't care about it. Okay? So now let's look at the STED laser. The stimulated. Okay? So first, you need to put some sort of phase mask there.
So you get the hole in the middle?
So you get the hole in the middle. Okay? And then
similar deal.
What's a similar deal, although actually it's a little bit different. What does it do to the red light? It reflects it. And what did we say it's doing to the orange light, which is actually, it's passing through. Again, it's wavelength dependent. This is sort of acting opposite to this, in that this is bouncing short wavelengths, and long wavelengths, it goes through. This guy is bouncing the long wavelengths, the red, and letting orange through. Orange is slightly bluer, shorter wavelength. Okay? So does that seem right?
Yes. Yeah.
And the difference is that with the red, you're coming in with something like this power, and with the green, you're coming in with the normal power. And so together, the only thing that is really fluorescing is right here in the center. And molecules that are exposed to this light, because it's such of a strong light, it comes back down. It lasers actually at that same wavelength. Yes?
So it only can detect one fluorescence at a time?
Okay. So,
That's
thank
really
you.
Good. Thank you. That was a really good.
Yeah.
Thank you.
Thank you. Was that, Greg, was that sort of meant for like both of us? That,
Yeah.
Now, there's a really good question. How, before, with the storm or the palm, it inherently was a single molecule technique, because we took, there was a bunch of fluorescing molecules, though they were all silent, and then you had to very gently excite occasional ones. Is there anything that says that you have to be at the single molecule level? And the answer is no. You can have a whole bunch of fluorophores in here and a whole bunch of, in here. There'll be stimulated emission over here, and the remaining thing, you can have one or you can have more than one. That's the main difference. This gets super resolution on multiple fluorophores. So potentially, this technique, you can get more signal out because you're not looking at individual ones, and therefore you can get faster time resolution. And in fact, that is the big difference. This gets faster time resolution. They can go to like 30 hertz with this, where with this one, you don't get to 30 hertz, you get, to 0.01 hertz, one image per, per minute or something like that. So in that sense, this is really great. The problem is what? What does it cost you to get this really good time resolution? Does anybody know? Money? Yes. Seriously, it does cost a lot of money for this. But, okay, let's say you got, you're Mr. Howard Hughes or something like that. So what else? You need to hit it with enough power over here to actually stimulate the molecules to come down. And that's non-trivial. You inherently need a lot of light. So this has to be a really powerful laser. In fact, it's so powerful that at first you think there's no way you can do it. In fact, you can do it with only certain fluorophores, because when you hit it, you have to make sure that you don't hit it and vaporize everything. And in fact, that is the main criticism of this technique. Yes?
I must be missing something. If you can have the phase mask to block out that little hole,
yeah.
Why can't you just do the opposite of that on the green laser?
You just make it small. You can't. Because you can't, because the laws of diffraction tell you that the smallest this can be is lambda over 2. And though those same laws don't apply to getting a small little hole. Yeah. Okay, Josh.
How do you know where to put the hole?
How do you know where to put the hole? Essentially, what it does is it sets up like a standing wave. Like in the STED laser, you have. Can I have the right? If you have something like this, the answer, the result is no light. And that's exactly what they do. They make sure that it's perfectly, no. This is twice. And then this is out of phase, and these two destructively interfere. So you.
Why didn't, or how do you know you put the donut hole right on top of the emission source rather than way out here?
This is actually coming from a laser, so everything is coherent, right? So the light coming here, there's a ton of light, and they all have a well-defined phase. And then what they do is right at the center, they cause a lambda over 2 shift.
But the exploration of that center needs to be determined somehow. Because we have to.
Yes. Yeah.
How do they do that?
That I don't know. Though apparently making this phase mask is a standard photolithographic technique. I don't know. Okay. Yes.
So can you play with the size of the phase mask to get a smaller PSF?
No. Pretty much this phase mask is sort of set. And so the effective size of this hole is set. The only thing you can do is you can turn up the intensity or down the intensity of this so you get more or less stimulated emission. And in general, the idea is you turn it up and the effective area gets smaller and smaller. But you have a problem of turning it up. It ends up doing nasty things to the fluorescence. Okay? Okay. And the bottom line, can I have the lights? The bottom line is you get a nice 35 nanometer instead of 200 nanometers. Okay. So we have 5 minutes before our break. Now I'm going to talk about electron microscopes, which is the standard way of getting lambda over 2, where lambda is much smaller. In particular, you have the light microscope. Essentially, the same is true for a transmission electron microscope. This is a transmission visible microscope. You have a light here, you have a specimen, you collect the light, and you detect it. Exactly the same thing is a transmission electron microscope. The only difference is instead of a regular light source, you have what is basically a heated filament. You heat the filament enough, and then you accelerate it enough, you will have a light which is at very low wavelength. So you hit it, and then you detect it. That's one way. That's called a transmission electron microscope, and it gets very good images. Very good, meaning really high resolution. There's another way that you can put instead your detector here. You can look at the scattering. And this is generally called the scanning electron microscope. It gives you really neat pictures. Okay? Now, in fact, so the images, in this case, this is using scanning electron microscope at various magnification. You can see that you can, this happens to be a bacteria, so that's about 1 micron by 3 microns. And this over here is an individual DNA molecule with this size is 200 nanometers. So you can see just a few nanometers. Okay, so let's actually figure out what the resolution, what is the lambda. And so you get lambda over 2 if you have diffraction limited resolution. So who's that guy? Wow, you guys are like fast. Okay. Okay. What they have is, remember, they heat up a filament, and then it gives off a few electrons, and then they put a voltage there. So essentially that's like a voltage is like the positive charges, and the electrons then get sucked into it, start moving rapidly, and then they hit the sample, and then you look at the sample. So how do we determine what the wavelength of these are electrons? How do we determine? Remember, you got famous. This is sort of the beginning of quantum mechanics. So what is it? What's the relationship between lambda and energy and momentum? Yeah. E equals h nu, which is hc over lambda, and P is equal to h over lambda. And h, where does the Planck constant comes from? That's what h is. Anybody know? It comes from the law of blackbody radiation. That the electromagnetic radiation emitted by a black body, that absorbs everything, and what does it do? It turns out it radiates it. It can be modeled as a set of harmonic oscillators with quantized energy of the form E equals h nu. This was a big question. The classical theory of blackbody radiation looks like this, and in fact, they looked at stars, for example, which can be treated as a black body, and they look like this. And it turns out this was the beginning of quantum mechanics. Okay, from this, they got what H was. And again, our goal is to get what lambda is for these electrons. So how do we do it? We know what H is. We know what C is. We don't know what lambda is. Do we know what E is? Okay, yeah, what is E in this case? What's that? QV. V being the voltage. So, okay, generally you have, let's say, 100,000 electron volts. So what is lambda? And H is this value. Okay, you guys got to do this in a hurry because you have to go on break. But you can't go on break. Oh, look at that. People like reaching for their whatever. Tell me what lambda is before you go. So what is lambda? Oh, look at this. He says, I don't have my calculator, and he says, I do, but I don't really feel like it. Okay, he doesn't allow, he's not allowed to get up. Come on. I want to know what lambda is. Anybody has a calculator, either on the computer or whatever? Yeah. Sorry, guys. I'm just can't let you go. It's not my fault. Oh, it's amazing. All of a sudden, he got out his calculator. Come on. He is HC over lambda. Therefore, lambda is equal to HC over E.
I guess 11. Oh, I guess I'm going to do minus 10. That's probably 10. JK. It's really fun.
Does somebody have the answer?
Oh,
yes.
Because I used...
Shh.
I don't know if I got 0.0123 nanometers.
Ah, okay. Let's see what I got. I don't know. You got point what?
0.0123.
I don't know. So we're a factor of 3 difference. I don't know what it is. But the point is, it's some really short number. So you should be able to see lambda over 2. So you should be able to see, 0.02 nanometers. So that's 0.02 angstroms, where the width of a hydrogen atom is like 1 angstrom. So can you really see this kind of resolution? No, you can't. So what's the problem? What's the problem? Come on. Guys, don't you want to go on break? Lambda over 2, if the thing is diffraction limited, is your resolution. That's it. But lambda over 2, it says we can see unbelievably short. And that's not true. So what's the problem?
Must not be diffraction limited.
Must not be diffraction limited. Hey, man, let's give him a real. There we go. There we go. It's not diffraction limited. Why isn't it diffraction limited? Because the so-called lenses that you use to make the image are not at all diffraction limited. That is, in fact, the resolution is something like, 200 times what it is if it was diffraction limited. So the resolution is more like 1 nanometer. So the EM are far from ideal. Whew. You have about 2.5 minutes. Get up, go. Power nap. Okay, time to power nap. Oh, whoa. No, you don't get to come in at yeah. You get 10% off.
Yeah, no, that's fine.
Yeah.
Better than zero. What is that?
That's the
Why is that for?
because you need to find a little better.
And you use this PC. H over Lambda. Which is how we're solving for Lambda.
Linton is H over Lambda.
Essentially this equation. So I solve for Lambda. Well, we know the
We know it.
yeah,
The voltage and the Is 1,000 a voltage or is it energy?
It's kilo electron volts. So yeah, that is a voltage.
Yeah, so I think you just multiply the voltage by
Charge in the line.
yeah, that's what I was doing. 1.60 to the minus 19 multiplied by if you get 1.6 times 10 to the negative 14.
What's the charge in the electron?
1.6 times 10 to the negative electron volts.
How many electron volts do you
Or
have?
sorry,
Coulombs?
I think I'm like mixing stuff up for you now.
But then,
Unless the 100,000 is energy.
yeah, because H is given in terms of electron volt seconds.
Okay, so let's do this as 100,000 as the energy, and we can take the energy divided by H, which we knew, which is not anymore.
4.1 times 10 to the
And that was in terms of electron volts.
Doesn't seem like much of having a power nap.
Yeah.
Okay, you got to stay awake for the next 15
I
minutes.
think what you need to do is give us like a break so we can figure out the question faster because our brains are all shut off. You know, it's like I can't work right now. Whereas you're like, yo man, you gotta work. I was like, I need sleep, man. That's cool. I'm getting weird ass numbers, dude.
Okay. So now we get to at least the fun part of some really cool images. So this is for a transmission electron microscope, and basically, the resolution is on the order of 1 to maybe 5 nanometers or so, and again, it has to be a very thin sample, so the electrons would hit it. And it won't be all absorbed, that actually you can have something to collect. And so, you can see mitochondria, you can see the nucleus, you can see the DNA, and this here is a couple of bacteria. Come on, you can do it. And this is DNA replication. You can see an individual DNA. Okay, and now this is like whoa. So this is a scanning tunneling microscope, and some artist, it's not in color. So this is, somebody's imagination. So this happens to be a cat flea. I mean, check that out. Is that not cool? And I don't know actually what the one at the left is.
It's like part 3S.
Yeah, I know. I can read that.
That's terrifying.
Yeah.
This is a fly. This is a
croissant with little baby fangs.
that's amazing. The head of a human flea. Man, he's like.
The croissant looked like paper mache.
This is a human eyelash. Somehow making it green. I don't know.
Beautiful.
Oh, well, that's it for that. Okay. In the last 10 minutes, I could give you it off, but I won't. Yeah, there'll be another 10 minutes before you can take a nap. I want to talk about the advantages and disadvantages of electron microscope and how combining them really can give you some amazing results. Okay, so electron microscope is great. You get molecular type of resolution. You can even do in vivo by going to very cold. Because what happens in electron, yes, it's forming an image, but an electron is charged. And so something that's charged going through this biological material is not very nice. And so you tend to get a lot of damage. So what everybody tries to do, well, first is you fix the sample, which is basically means you're looking at a dead sample. And you also always try to go to minimum of exposure. Another alternative is you go to very low temperatures, like liquid helium or liquid nitrogen. And at that temperature, things tend to be very still, and their interaction with electrons, while still there, tend not to be quite as damaging. Okay, so that's called the cryo-electron microscope. However, even then, you don't get atomic resolution, and for sure, it doesn't work on live samples. And X-ray crystallography is really great. That is, you get atomic resolution. However, with X-ray crystallography, it's highly artificial in that in order to form the crystals, you need to put it in, all sorts of strange buffers and so forth. And of course, there's no way of doing it in vivo. However, if you take cryo-electron microscopy, where you can't do live samples. That's for my kids in my class, in my graduate kids. That will be their job, is to do this on live. So okay, it's not live, but nevertheless, you can take cryo-EM in a cell, get an image of it. You don't have quite molecular resolution. However, if you then take the X-ray crystallography, which is done in these weird buffers, and then fit that to the overall image that you get with cryo-EM, you can actually get an image of the thing in a cell. And I'm going to show you one such example of this. Okay, so the recent revolution is doing cryo-EM. What happens is you have an image of something, and you take an image. There's inherently a lot of noise, so they do all sorts of image processing, which I won't really go into. They manage to get three-dimensional reconstruction because sometimes, it's here and it's here and here. They separate them, get the average, and they say, okay, average, let's say, and the X is there, and then the Y is there, and the Z, and they can do it. And so this is what I consider the most amazing thing. Remember we talked about the F1, FO ATPase, how basically your body converts ADP plus phosphate into ATP, or vice versa. They imaged, this is a single F1FO ATPase, and remember, the question is which one is FO and which one is F1, I'm not sure, but there's a stalk between them. This is a stalk. So they got this image from EM. In X-ray crystallography, they managed to recreate or to crystallize this guy and this guy and this guy in artificial conditions, but they managed to overlay those onto the entire structure. And you can see that in general, actually, it's very good. It's surprisingly good that the X-ray crystallography apparently works. And so this is just amazing. Amazing. Okay.